Welcome to IME's free GAMSAT webinar 7. This is a special webinar because the GAMSAT is only 9 days away. I know a lot of you are probably very stressed at the moment, and I have received lots of feedback from lots of candidates in the past that can't make it today, but are definitely going to be watching the recording. It definitely is, well, it's 2023, first GAMSAT for the year. It's a lot of people will be seeing the GAMSAT for the first time. It's definitely going to be a new experience for all of you, but it's a great experience. The best advice I can give you before we get the ball rolling today is that the best practice you can get for the GAMSAT is to sit the GAMSAT. So a lot of people can definitely attest to that. The reason we say that is because the GAMSAT isn't just what you know, it's how you do under pressure, time pressure, and how you can deal with your nerves and how you can prepare for what is a mammoth exam. Today's session is going to be, I mean, they're all very, very beneficial. We've had 6 in the past. It's amazing we're already up to number 7. We're going to be discussing interpreting commentary pieces and some ever so deceiving Mendelian genetics. I know a lot of students tell me that, oh, it's fine, Mendelian genetics is very easy, and they skip it. But I think hopefully after today's session, you'll get an appreciation for the complexities of Mendelian genetics and the different types of Punnett squares you can derive, and trying to read a whole block of information and designing your own Punnett squares. So hopefully today's session will give you an idea of going back to your study and going back to your notes and making sure you understand what certain molecular terms mean or biological biochemistry terms mean, because it will definitely help you in the future when you're trying to approach these Punnett squares and these genetics questions. So if you would like to discuss your GAMSAT scores, your possibilities for entry, or your or just any pathways into medicine, anything like that, you can message me at the institute. We can have a private consult, or we can discuss your pathways, and it is free, it is confidential, no one will know. So we're here to help you at the Institute of Medical Education. So please, if you're lost and you need any support, just shoot us a message. I'll give you more information in the next slide on how you can contact us, but just note that we can support you in any way if you do require help. So these sessions are recorded, just a reminder, and you will receive a link to access this and previous sessions via our mailing list. So we do post these on our website and via YouTube. So if you are watching the recording sometime in the future via YouTube, if you do want to be part of our mailing list, again, contact us and we can put you on the mailing so you don't miss out on any updates and any other material and free material that Institute of Medical Education releases. So, as I said earlier, you can follow us on social media. So you can go to our website, imemedical.com. We have a lot of free stuff there, free essay generators. We have free exam, interview generators or question generators for interview. You can follow us on Facebook, YouTube, LinkedIn. If you search Institute of Medical Education or our handle, I think it's at imemedical.com. We regularly post free material, whether it be questions, solutions to ACER questions for different exams, and you can definitely, I mean, we do look at all your, there have been lots of comments recently on our videos. We do respond to all your comments and all your queries. So if you have any problems with your ACER material or anything like that, you can definitely shoot us a comment and we can help you answer the questions that I guess you just won't get via doing the exam yourself. So you can follow us on Instagram at pre-IME, at pre_IME, and TikTok at imemedical.com. I'm sorry, at IME Medical, where we go through, well, I guess it's pretty lighthearted, and you get small snippets of which I guess are definitely high yield information of different topics, whether it be chemistry, physics, biology, or the humanities. So if you are interested in any of our free material, just follow us on social media, or you can come meet us in person here at the Gold Coast in Benoa. We're about 5 minutes from Surfers Paradise. I'm here at our beautiful building, brand new, great facility. You can have a chat with us and a consult if you like for all your medicine or dentistry or optometry needs. So today's class. So we're going to first take a look at section 1, commentary and opinion pieces. And then we're going to look at Mendelian genetics for section 3. And finally, address any of your questions, and we can discuss any tips or anything you should be doing, I guess, in the last 9 days for your GAMSAT, and how you should be approaching the last few days before your GAMSAT. So let's begin with section 1, commentary and opinion pieces. There's going to be 2 questions in this set. Jeremy, are you there?
Yeah, I'm here, Hassan.
Yep. So we are going to have 2 questions in section 1 of the GAMSAT. So in 2023, I think it's 62 questions in 100 minutes, and it roughly translates to about, I'd say, 1:30, 1:40 per question. There is no reading time or perusal time in this exam. It's all included in the time. So you just start 100 minutes from when you, so I think section 1 and section 2 are done together in a block. But the point is, there is no, you have 5 or 10 minutes reading and then you start. It's all included now. So I'll give you, how does 3 minutes sound? Is that okay, Jeremy?
Yeah, I mean, 3 minutes fine.
So yeah, so for those playing along
minutes.
at home, you can give yourself 3 minutes. I mean, if you want exam conditions, if you don't want exam conditions, take it at your level. But we'll give you 3 minutes, and then at 1:30, I'll transfer to the next question. And then we'll, the good thing is, because we've got the poll here, actually, Jeremy, you can jump to the next question if you like via the poll. But for those playing along at home, at 1:30, I'll change to the next question. So I'll get my phone out, just the time. Okay. If you're watching this recording at home or from wherever you are, you could be overseas on a nice holiday, sipping your pina coladas. You can come back in 3 minutes, or just fast forward 3 minutes, and we can go through the answer. Are you ready, Jeremy?
Ready.
Okay, your 3 minutes starts now. Okay, 1:30 is just about up. Final question. Okay, so we have 10 more seconds. If you can just fill in your poll answers as well, just so we can, it's academic at this point. So 3 minutes is over. So see how you went. Fantastic. So just before we dive into your responses there, Jeremy, and to those playing along at home. So today's topic is about gender inequality in biomedical research. Yesterday was International Women's Day, which highlights obviously the great achievements and the effect of women in the workplace, and how they benefit society in every facet. The world is a better place when women are more involved and engaged. I think about it most times when, if you look back in, so even you have to go back to 1900s, 1800s, before then, when women's views were not valued, and how much it held back the progression of humanity by not taking into account women's views, and they're incorporating them into everyday life, workplace, and that idea of equality, and how much women do mean to society, and their contributions should be definitely respected. And obviously there should be International Women's Day every day, but it's nice to at least let those around you know that women's contributions should be respected. And there's a lot, it's not over. There's a lot more to do to fight for equality, equal pay, equal respect in the workplace. There's a lot more we can do. And it doesn't just, it's a shame that it extends to medicine and research, but hopefully we can do a lot to make strides in this field. So we've got 5 passages here regarding gender inequality, and they're either going to be, I guess, pro or con gender, this idea of gender inequality in biomedical research. So we've got 5 here. I'm not going to read through all of them. You can pause the screen and read through all of them. But the first question asks us, the first question says, considering the 5 extracts presented, how many oppose the notion that women are overlooked in biomedical research? 2, 3, 4, or 5? Now, Jeremy, how did you approach this question? Can you give me your thoughts?
Oh, well, I thought it was tricky because I think I took the more difficult approach and tried to answer the question straight away rather than like changing the question a little bit. Like there's one tip I remember now, like if you have a question that asks what is not something, then it's easier to first answer what is something. And then, so it's like, I don't know, not a double negative, but trying to juggle 2 different thoughts in your head at one time. Like first trying to answer, is this talking about women overlooked in biomedical research, and is it opposing that notion? So maybe at the end I was trying to go first is what is talking about women being overlooked in biomedical research. And yeah, so I found the comments were actually pretty challenging, and like whether there was a common I was trying to find a common theme.
It is
Yeah,
tough under time pressure. Hey, you have to read that block of text and then come up with an answer as well.
So they're all kind of different in their own way. But then when I saw the second question, I thought, oh, okay, I guess they're all maybe talking a bit about it, trying to be more factual, and we've got ratios and statistics and now requirements. So they're all. So that's why I felt like the second question was on facts. But yeah, this first one, I thought, okay, the current ratio of women to men 2 to 1, that doesn't overlook women in research. So I thought that was, I guess, a ticking that box. So That-
So does this, would you say this opposes or does this support the notion that women are overlooked in biomedical research?
This opposes the view that women
Okay,
are overlooked. Yeah.
so we can say that this
So,
opposes, yep.
yep. Number two, we had well, it's saying that we're kind of, well, see, this is hard. Like, are women overlooked for drugs, for pregnant women taking drugs?
So
Well,
what's the
then
crux? So what's the crux of this question? So this
I
statement,
guess,
what's the crux?
it could also be talking about, it could also oppose women being overlooked because if they weren't overlooked, would they also do the same studies on pregnant women? Or yeah, is it like, are they overlooked because they aren't participants, or are they not overlooked because, you know, there's health risks for them being participants? So that was challenging.
So let's think about question. So if we think, let's go back to one. So one is saying here that this comment is saying that women are usually excluded from clinical trials, and they're arguing, well, it's a popular and tenacious view because women actually make up 2 to 1 of the funded trials. So this comment here is trying to say that women aren't excluded from clinical trials, they actually make up the majority. So it's
Yeah.
saying that women aren't overlooked. So that one's correct.
Yeah.
Number
That one
two
was
is
clear.
saying that it's very hard to give women drugs today because if pregnant women drugs, there's a lot of the new drugs don't, and they're not approved to be tested in pregnant women because they're not included in clinical trials. So is that kind of saying that pregnant women are represented in clinical trials, or are they not represented in clinical trials? Because they're saying a lot of the drugs that are given to pregnant women today, they're extrapolated from data in clinical trials of men. So is that saying that women are being treated correctly in clinical trials or treated incorrectly here? So in terms of trying to give a drug for pregnant women, but we're doing clinical trials in men, how does that compute?
Yeah, I guess they are overlooked because you could probably, you know, you could still, I guess you probably make a safer way of testing it on pregnant women, maybe smaller doses. I don't know.
I guess the idea is, I mean, it comes back to the whole respect for women. You know, it was International Women's Day yesterday, and the fact that we're in 2023 and we're giving pregnant women medication based off clinical trials in men is ridiculous. And the fact that
it's like, if we never conduct any research, then we're giving, like, it's probably true that they're always going to need some sort of medication. You know, it's not like they become a different than the rest of us once they're pregnant. You know, so they're going to need a medication, and so it's better. If we don't want to overlook them, we should probably also include them in other studies.
It's this idea as well that, oh no, pregnant women, their hormones and their body changes during pregnancy, we can't do clinical trials on them. It's like, you can. I can guarantee you a lot of pregnant women would be accepting to do it if they know that it's going to make the lives of other pregnant women in the future better off. Because, you know, it's something that's been lagging from the past. This idea, no, we can't test pregnant women. Have you asked women? Have you asked pregnant women? You know, it's this idea we just assume that's what they want. How about you talk to the actual women? So that's the idea. What about option 3? So option 3 is about saying general perceptions have taken priority over data. There's now a requirement that gender must be considered in every grant application involving human subjects. Does that support or oppose the notion?
That opposes the notion they're overlooked.
Definite.
So is it?
Oh, sorry. Yeah. Sorry. Tick. Sorry.
That'd be a tick.
Yep.
Yeah. See,
it gets confusing now because
it's confusing.
Yeah. This is what GAMSAT does to you. So we're trying to look for the ones that oppose, those are ticks. But that doesn't necessarily mean it's a good thing. It's just the question is asking for those that oppose. So why does it oppose the notion that women are overlooked in biomedical research?
Well, I guess it's like saying that they're saying that there's now like having a requirement to state gender means that no one will be overlooked,
Mm,
you know. So that's, you know, but
that's
I
true.
guess, but then
That's true.
saying that and but I don't know if that's related to the question specifically. I think the angle would be more like we're putting in this requirement because women were overlooked, and that's why there's a requirement, and so that it's, well, maybe actually it's in favor that women were overlooked, and so now there's a requirement.
But if you read the first sentence, the tone of the first sentence tells you that this person is probably ticked off, like they're annoyed that people keep telling them that women are overlooked. But how are women overlooked if they're considered, there's gender requirements in every grant application? They're not overlooked. That's their argument. Because the idea is that they're arguing that general perceptions have taken priority over data. You might think that women are overlooked, but the data is telling you that we have to include women in these research. So we're not actually overlooking women. So you can tell by the tone that they're pretty annoyed, just by the first sentence.
Yeah, okay.
So they're arguing that general perceptions have taken priority over the data. The data actually shows that gender must be considered in every grant application.
That's true. And yeah. And it's like what's happening today. It's not talking about the past. The question isn't asking about the
You can
past.
tell this person, comment 3, because when I got this information, I got it from Science and Nature. There was lots of commentary articles and people giving their voices about what they think. And I mean, with this is kind of like a section 2 question, right? With the essay writing, you can't assume that everybody agrees for that gender inequality doesn't exist. There are people out there who believe that gender inequality doesn't exist. And you have to understand their rationale. You might not agree with it, but in order to argue against someone, you have to understand where they're coming from. So this is what their arguments are. And this is what makes your section 2 essays a lot better. Because if you can acknowledge the other viewpoints and acknowledge their sound arguments, you can therefore say, okay, I acknowledge your arguments, but let me show you why you're wrong and give them evidence and proof. And that makes your essays a lot better because you've taken into consideration the opposing view and shown them that, yes, I acknowledge your view, but you're wrong. And you explain why. So that's how you can improve, not just section 1, obviously it's section 2 essays. It's this idea of understanding the opposite. It's not, you can't just live in this utopian world where everybody agrees that gender inequality exists. There are people out there who don't believe this. And these 2 viewpoints are definitely highlighting this. What about 4? What do you think about 4?
So the first line says that there is inequality.
Yep.
So that would say that women are overlooked because it's male bias, although we identified female bias in studies of reproduction. So
what's this sentence saying then? 75% of studies did not specify whether the animals were male or female. Why is that an issue? Well, it says here we identified female bias. So generally speaking, do you think people would be using, I mean, look, you're not in medical research, but the general consensus in research is that it's easier to use male mice or rats than female mice or rats. Because obviously the females, they have their own, reproduction, fertility. There's a lot of whole other factors. Whereas with the males, it's just you have a male, You do your experiment, you don't have to worry about the fertility period, changes during fertility periods, hormonal changes, X, Y, Z. It all comes back. But again, I'm saying this, it's a stereotype. It's not something that we believe in, but that's a lot of people just say, let's just use male mice. It's easier to work with male mice. That's the stereotype. Obviously, you're going to get people now in the comment section saying, what do you mean? That's not true, blah, blah, blah. I'm not saying I believe in this. It's a stereotype. As someone who was in medical research for a very long time, I can tell you that that stereotype is still around. So what do you think this sentence is trying to say then?
It's trying to say there's, women, like the gender's overlooked.
Because it's like if
women
75% of studies generally don't say specify the sex of the animal, what sex do you reckon the animal is going to be anyway?
It's going to be male.
Going to be male. So the fact that they're not specifying the sex, I'm telling you, I've read a lot of journal articles where they don't specify the sex, and it really annoys me, because it's like, well, sexual dimorphism exists. So sexual dimorphism is where different sexes have, like for example, humans, sexual dimorphism in humans, women have wider hips for childbirth. That's a big one. Bone densities are different between males and females. So it's the same thing with mice. There could be sexual dimorphism between the sexes, changes that are sex specific. So this is why you have to, if you're doing research, do studies in both males and females if it's a generalized disease. If you're looking for testicular cancer, obviously you use male mice. But if you're looking for diseases of, say, diabetes, you've got to use both male and female mice, because they might behave differently and they might have different results. So that's why it's important. Now, the last one says sex disparities in the experience of disease are hardly surprising given the fundamental biological differences between men and women. So what's that saying? Is that supporting the notion that women are overlooked in biomedical research?
No, I don't think so. I think it's taking into account, the gender, sex differences. So I guess it's.
But if it's
doesn't
acknowledging
oppose the
that,
notion.
but if it's acknowledging that, doesn't that mean it is overlooked?
I think it's opposing. I think it's a tick.
Well, let's read it again. It says sex disparities in the experience of disease are hardly surprising given the fundamental biological differences between men and women. So what this is trying to say is, it's kind of a comment saying, well, you know what? Diseases, disparities in what we're seeing in diseases, it's very obvious because men and women are different. So it's trying to say that it supports the notion that women are overlooked because there's sex disparities in the experience of disease. It's this idea of, in biomedical research, remember, this is in the context of biomedical research. So why are we seeing all these disparities in the diseases? Why are we seeing all these disparities, but we can't find cures? You know why? Because we're not doing experiments to look into women's health as well. So it's kind of acknowledging that there is this problem that women are being overlooked. So this one is an X. But look, the good thing is, Jeremy, you were correct. You said 2, so you were correct. That's all that matters. You don't have to do this in the GAMSAT. You got it. You move on, because once you look into a question too much, you start doubting yourself. Another tip for the GAMSAT I have for you is, if you pick an answer in a minute 30, write it down, move on. Don't overthink it. You have no time. Just move on. So you got 2. You're correct. But look, in the GAMSAT, this is what's going to happen. You were going to narrow it down to either 2 or 3. The answer was A, but it's a 50-50 at that point. So you had a 50-50 chance. This was tricky, but based on the context, you'd understand that it's definitely supporting the notion. So there's 2 extracts that oppose the notion that women are overlooked in biomedical research. So those are the ticks. And there are 3 that support the notion that women are overlooked in biomedical research. So those are the crosses. And it comes down to, quite literally, in the GAMSAT, you don't have much time, right? So it really does come down to how good you are at reading and critically analyzing what you read. And that comes with practice. and how good you are at discerning subtleties in text. So that leads to question 2, which is, according to the passages, the opposition argues that gender inequality in biomedical research is primarily based on facts, shrouded in fallacy, in favor of men or in favor of women. Now, what are your thoughts on this, Jeremy? So we discussed the opposition. So the opposition is 2. Oh, sorry, not 2. So the opposition is 1 and 3. Not laser, sorry. Come on, pen. Where'd you go? There you go. So opposition is 1 and 3. So what I'm going to do is I'm going to highlight.
Well, I didn't think it was fallacy. I thought, yeah. Well, at first I thought, oh, maybe it's in favor of men, and then I was like, well, women to men was 2 to 1, which seems like it's in favor of women. And then, then the other comments were saying otherwise. So I thought the closest I could get was based on facts. That was where I was, leaning towards.
Because, so we can definitely cross off C and D. We're left to 50% because the opposition isn't arguing, because you can see clearly they're not arguing that men or women. I mean, the idea is that, first and foremost, they're not arguing that gender inequality is in favor of men. No one's arguing that. I don't think anybody is arguing that. I mean, realistically, yes, there's a small pocket of lunatics, loonies, I'd like to call them, who believe this, but they don't represent a statistical significant amount. I say that, and then you go to the elections and you see, it depends where you're from. In Australia, thankfully, we have checks in place to make sure this doesn't happen, because education is available to everybody. But in some instances around the world where education isn't provided to everybody, those views could be present. Obviously, we still have education in this country, and there's still people that fall through the cracks. But look, generally speaking, not many people are arguing that men of being, I know you can, I'm not going to start dropping names, but obviously in this day and age, there are people here who are trying to perpetuate that men are victimized, but that's far from the truth. Men are not victimized. Like, please know your privilege. Let's put it that way. So definitely not C. In favor of women, I wouldn't say that's true, because they're not arguing that gender inequality favors women. They're acknowledging that women, just by reading the context, again, you have to understand the context it was written in. They acknowledge that women, just by reading, they acknowledge that women are being treated indifferently, but they're not saying that it's not saying that gender inequality is in favor of women. That's not what they're arguing. It's either based on facts or shrouded in fallacy. Now, if we take a look at the first sentences of 1 and 3, it says, women are usually excluded from clinical trials. It's a popular and tenacious view. And obviously here it says general perceptions have taken priority over data. So what do you think the arguments are then? Are there arguments that gender inequality is based on facts, or because it's just fallacy?
Yeah, like I was thinking their argument is more based on facts.
Yeah, the opposition is arguing facts, but what are they arguing that so the opposition to the opposition, they're saying that they're making these baseless arguments that women are excluded from clinical trials, but no, there's 2 to 1. General perceptions are taking priority over data. No, that's not true. There's a requirement now for gender, for gender in grant applications. So they're arguing, what do you mean? You're saying all this.
That makes more sense. Yeah,
random stuff, and it's not making sense.
They're saying it's like the gender equality then is fallacy.
Mm-hmm.
Yeah.
So they're arguing that your argument. So if I was someone who supports if I'm someone who's supporting the notion that gender inequality exists, the argument for, if I'm arguing with somebody who says, no, it doesn't exist, they'd be saying, well, the reason why it doesn't exist is because you're just ripping out all these random information, but I've just proven you wrong. You're saying that women aren't in clinical trials. Women make up 2 to 1. You're saying that you're telling me that women aren't represented in grant applications. That's not true. In grant applications, you have to have gender quotas, so these sorts of things. So they're saying your argument's fallacy, but I'm telling you my argument's data. So that's it. So do you have any questions on these two questions before we move on to genetics?
No, I think it's it's a good example of questions having different, what is it?
It's kind of like you have to read the question again. This is section 1 in a nutshell.
Questions can have these different perspectives, and you can
based on who is asking the question.
Multiple ideas to fold up at once, and so if you can kind of get rid of a few, and it'll open, like according to the passages, well, that's there's not much else we can go off there, so you can forget about that part, and if you can then just keep in mind the what just narrowing it down to the opposition argues, so just those two, and then it's gender inequality. So it's if you can kind of narrow it down so that you're just thinking about one idea, where there's double negatives, if you can change the question so that it's easier to grasp. It's just doing the practice, I guess.
That's right. Yeah, I think that a lot of people struggle with section 1. The stereotype is that you can never improve your section 1. That's not true. It is difficult to improve your section 1. You can improve your section 1. It just requires a lot of the 1%ers. The 1%ers are, you have to read, not just read, actively read. What are you reading? And try to extract important information. You can read a whole book and get nothing out of it. That's not the idea. The idea is you got to actively read. That comes with practice. Being able to read a large amount of information and extracting the important information comes with practice. So there is no course. Honestly, there is no course that's going to help you improve your section 1 overnight. It comes with practice. It's kind of like I give a description, the example of music theory. So you can't learn, if you're trying to learn oral music theory, trying to hear sounds and try to be able to write them down, write down the music notes and the rhythm and all that sort of stuff. It doesn't come overnight. It takes practice. It's the same thing with section 1. There is no quick fix. There is no quick fix. You just got to practice.
One thing that helped was putting pen to paper, and then also trying to get a good way of mapping it out. Like I started first by going, should I do like yes, no, like first going, okay, let's put question, like answer 1, 2, 3, or like the statement 1, 2, 3, 4, and 5, and or should I put against each one an O for oppose, or F for or a tick or a cross. So how am I going to actually, like the what do you call it, operationalize answering this question?
Don't keep it in your head. If you keep it in your head, you're lost, because in the GAMSAT you just have too much pressure on you. There's too much going on in your head. You do get given, I think it's 2 pieces of paper now. I can't remember. Asa will tell you. You get given pieces of paper because you're doing your exams, obviously in one of their rooms, wherever around the world.
2 pieces of paper, and I found that 2 was good for section 1, like it's easy, but the section 3, I really used every corner of every inch of the 2 pieces of paper. So I learned to I'm trying to practice writing smaller or organized, so I just take up a small amount of space.
It's annoying because I know for example, if you are sitting the UCAT at the Pearson VUE rooms, they tend to give you a laminated piece of paper to write on. So that's a big problem. So we've got a new member entering as well. Great, just in time. Wait for Monica to attend. Hey, Monica, are you there? I think your audio. Hey, Monica, are you there? Hello, Monica. I think you're just on mute. Are you there, Monica?
Yeah, hi, sorry.
No, you're right. I'm just in time for section 3. We just went through a section 1 stimulus, but don't worry, it's recorded and you can have a watch of that when we release it, I think next week.
Okay,
So
awesome.
don't worry, but you're just in time for section 3.
Sweet, thanks.
Are you sitting the GAMSAT for the first time?
Not the first time, but I am sitting it. Yeah.
Okay, so we'll hopefully this is some good practice for you. But we'll go through at the end some tips and tricks to get you ready in the next 9 days for the GAMSAT. Because I know obviously you're probably very stressed. I know everybody's stressed at the moment.
Yeah.
But we'll go, hopefully to this. So I purposely, when we design this question, to trick a lot of students. You're either going to get this or you're not going to get this. But the good thing is when we work through the answers together, I can guarantee you when you get into the GAMSAT, you're not going to make a mistake. So because Mendelian genetics is one of those that all students say, I get it. But then when they get given a question in the GAMSAT, the training wheels fall off. So we'll see how he goes, and then we'll go through the answers together. So just before we jump into the question, I'll give you some background information. We know that obviously with genetics, we know that there's 2 alleles that exist. I mean, it's this perception that only 2 alleles that exist. So that's one dominant and one recessive for a given gene. And these days, it's kind of a gross oversimplification. It's not just a dominant and a recessive gene for the alleles, right? So yes, species like humans, we're diploid, which means we possess 2 alleles for a given gene. So remember, sorry, one from your mum, one from your dad. So that's diploid. It's clear that many other variants of genes may exist in a greater gene pool. So you might have multiple alleles for one gene. I mean, we'll go through that later on, but just keep that in mind. It exists in a lot of the animal kingdom. I know in here we've got the Indian rabbit. I think I saw one, I think it was gene C. There's multiple genes. I think gene C had something like 4 or 5 different alleles for a specific gene for color. So that's what we're getting into today when we're looking at the phenotypes for the Indian rabbit. So when many alleles exist for the same gene, we generally use the convention of the plus for wild type, and that's considered the standard or the norm. And all other phenotypes or genotypes are considered variants of the standard, meaning that they deviate from wild type. So you've got wild type, and then you probably see in research papers or anything, you probably see a plus, which means it's this, you got one gene from the mum, one from the dad, that's the wild type, that's the normal, or you could have plus minus, which means you've got wild type and maybe the recessive allele, then you've got minus, which is like the knockout. So you usually see that on top. Maybe the gene is CFR or something like that, or CFR or anything. So you might see it like that. So if you don't understand that, if you're watching at home and you don't understand this, I suggest that you do probably go back quickly and have a look, because you never know, in the GAMSAT, they might chuck at you a question like this regarding these pluses, minuses, and as always with section 3, the more you know, the easier it gets. So there's a little 1%ers you might have to do to improve your score. But again, it's something to think about moving into your March or September GAMSAT. Now, getting into the question, we're going to be discussing Indian rabbit here. It's thought that a single gene, keep that in mind, controls for a general coat color, and rabbits have either brown or white fur. I think it was gene C, I can't remember, C or E, and there's multiple alleles. You can probably look it up on Google, obviously, after, or those playing along at home. It's pretty interesting, it's pretty neat, I think it's pretty cool. For rabbits with white fur, so we've got, it's hypothesized a further gene may be involved in coat color and may result in variants which include chinchilla, Himalayan, and albino. So chinchilla is black tip, white fur, Himalayan, white fur with black paws, nose, ears, tail, and albino, which is white fur. Not important for the GAMSAT, but not important for this question, But in the GAMSAT, they throw all this extra information in there to trick you up. So it's not important, but just note that you've got brown, wild type is brown, and then you've got the, I guess the recessive or the non-wild type, which is against the norm, is the white. And for the white, you can have 3 different phenotypes. So that's the first thing you need to keep in your head. We've got brown, which is wild type, and white. But white has an offshoot where it can be albino, Himalayan, or chinchilla. Something to keep in mind. Now, this is where the training rules fall off for a lot of people in the GAMSAT. What I'll do is we're going to have 3 questions. I'll give you, so generally for section 2, you have 2 minutes per question. So it's 75 minutes and 150 minutes, 75 questions in 150 minutes. So 2 minutes per question, there's 3 questions, I'll give you 6 minutes. But because this is going to be a bit tricky to understand at first glance, in GAMSAT, not all questions you should be giving them the same amount of time. Some questions you can blitz through them. Some questions you might have to spend more time. But just make sure that you make note of the time. What I'll do is I'll give you 3 minutes for the first question, then 2 minutes for the rest. So we'll give you 7 minutes altogether. So I'll start the poll and just have a go at answering the questions. And then when we come back, we can discuss, see your approaches to answering these questions. And then if you blitz through this and if you feel as though this is comfortable for you, it's easy for you, congrats, because these are the sorts of questions, because look, in the GAMSAT, certain questions are definitely scaled. These are the sort of questions that if you get correctly, you'll probably get scaled higher, because genetics is something that you either know or you don't know, and it's definitely a difficult topic. But if you do get it correctly, my goodness, you're already halfway there. Are you guys ready? So I can start the time. So 3 minutes for the first question, and you've got, for those playing along at home, we've got 1, 2, 3. So you can pause the screen either here, here, or here. For those playing along now, so Jeremy and Monica, I'm going to start the poll. You've got all the 3 questions ahead of you. So if you want to jump the gun, you can do that. But I'll start your 3 minutes for the first question and 2 minutes each. Start time now. Okay, we've got 10 more seconds on this question. Moving on now, the next two questions, remember 2 minutes each question. Okay, so we've got 2 more minutes for the last question. However, those playing along in our session, so Jeremy and Monica, if you do finish early, because I see it looks like you're both finished. Are you ready? So we can, for those playing along at home, you can keep going. But for Jeremy and Monica, are you both finished? So we can move on, or do you would like more time?
I'm finished.
How about you,
Yeah.
Monica? Are you still going?
Yeah, no, I'm good. Sorry.
No, that's all right. All right, sweet. So we can stop there. Obviously, for those playing along at home, you still have about 1.5 minutes. But so this is a there's a lot to unpack here because there's multiple things happening. This is where it gets tricky because you're getting told that this is what the population is in this part of the country. We've got the south, center, and north of the country, and you're getting told what the percentages are of the rabbits in these parts of the country. Then you're getting told 3 different theories as to what the genetics or the genotype is of these rabbits. So just quickly, south of the country, you've got 3/4 to 75% are brown. So when you're in the GAMSAT, you want to make sure you just make a quick note, 75% brown, rest are just white. You've got in the center of the country, there's just a bit more white rabbits than brown. So you can write white just more than brown. So just more brown. Sorry, my pen is very small. And then finally, in the north of the country, there's an even split of brown rabbits, albino rabbits, and other white. So it's all even. So you can just say even. That's the first thing you want to know. So there's 3 theories. Now, before we dive into the theories, I have a couple of quick fire questions I have for you guys to see if you understand genetics and different types of meaning. So if I were to ask you what is a gene, can you tell me what you think a gene is? So Jeremy or Monica, anybody can answer. What do you think a gene is?
I can go first if you like.
Yeah, sure, go for it.
I guess a gene is made up of a certain sequence, or it's a pattern of a certain amount of DNA that leads to the expression of a characteristic and a
Mhm.
visible, some sort of characteristic of a yeah, a
Yep.
thing,
So
a
yeah, a
living thing.
gene. Yep, a gene is a section of DNA that encodes for a certain trait. So yep, that's correct. So if that's a gene, then what's an allele?
An allele is a way of describing that there can be different genes. So
Mhm.
if it's kind of like yeah, it's hard to explain. Like there are different alleles. Alleles are genes. Is that right? I think, hold on.
Yeah, alleles are genes,
Genes.
but what? So if you get one from your mum, one from your dad, what does that mean? Are they the same gene? I mean, are they the same? I mean, they're in the same. They're encoding the same thing, but are they the same exactly?
Um,
So
they
what?
could be different. Yeah, could
Yeah,
be
so
different
they're
genes.
variants. So they're variants of a gene. So they're
Yeah.
variant forms of a gene. Because remember, you get one from your mum, one from your dad. If we all inherited the same exact gene as everybody else, there wouldn't be any disparity between humans. We'd all look the same. We'd all be the same. It'd be a very boring place. So alleles are variants of a gene. Now, what's a chromosome? The reason why I'm asking this is because these are when I've noticed in the past with students learning genetics, they skip straight to the Punnett squares, but they don't understand what they're doing. They're just being told to mechanically
Yeah.
quickly do like maths in their head, but they don't actually understand what it is. So what is a chromosome?
I was trying to come up with I like to come up with like examples or like metaphors. And
Mhm.
so I figured out one for the gene and allele. It's like, a tree is like a type of plant. And then that's like an allele or like a gene. And then, like a phytosporium is a type of tree. So you can have different genes, but each gene can also have different alleles.
Mm.
So yeah, kind of hard to think, oh, there's different genes. Okay, well they're all different, but they're doing this same. They're affecting the same trait. No, it's like
Mm.
different genes, different traits, but within each gene, like within each trait, it can be different itself.
Mm. So what does a chromosome have?
Chrom-
So what does a chromosome have?
Chromosome.
Mm.
Chromosome is like like different genes or different alleles are then located on what's called a chromosome, which
Yep. So
is
how many molecules of DNA is a chromosome?
it depends on the animal. I
Well,
guess.
well,
Does it?
one chromosome is one strand, so one molecule of DNA.
Oh yeah,
How do you
yeah.
pack that? How do you pack that as a chromosome? What do you use? What proteins are important?
Histones. Yeah, so you
Yes.
wrap the DNA around histones. So it's
Mm.
and
Perfect.
then that can be then condensed again. So it's like
So then how many pairs of chromosomes do humans have?
two.
How many pairs of chromosomes do humans have altogether?
Oh, pairs. How many
Yep.
pairs? Yeah, they have the 48 or 46.
46. So you got it.
46.
So sorry, 23 pairs, 46 chromosomes in total.
46 chromosomes. Yeah, 23. Unless you've got the trisomy.
Yep, unless
You
you've
know,
got the trisomy or the monosomy.
yeah, where
So that's
you've
good. So what's trisomy?
one you might have 3 chromosomes within one group of like one chromosome might have 3.
Yep,
what
so it's
it's
an extra chromosome. What's the best example of trisomy that people always jump to when we talk about
Is it
trisomy?
Down syndrome?
Yep,
Is that
so
the
it's generally the presence, yep, of an extra chromosome. I can't remember which chromosome it was
is it
actually. Should have wrote it down.
21?
Could be, could be. What about monosomy? What's that?
Monosomy, again, it's mono is 1, so it's where you're missing a chromosome.
Hmm. So there's examples you could get in the GAMSAT. I think is Turner syndrome. So it's females born with only one X chromosome. So we know obviously sex chromosomes. We've got female, male, XX, XY, and chromosomes come in pairs. Humans are haploid or diploid?
Diploid.
Diploid,
Guide
yes.
for 2.
Yep. So because we get a copy from our mum and a copy from our dad of the gene, right?
Yeah.
And finally, which sex cell, so the gamete, determines the gender? I have to say that correctly, obviously, in 2023. But which one, biologically speaking, determines the gender of the child? Is it the sperm or the egg?
Oh, it's X and Y chromosomes. So it's the
So female
I guess
ovaries
it's
are
the egg.
X and Y.
The female. So
Remember, so remember, gametes
female are double
haploids,
X.
right?
Yeah, females XX, males XY.
Yeah, but like female, so sex gametes, are they haploid or diploid?
They would be haploid.
Yep, so they've got 23 chromosomes, which means they either have an X or a Y. So what about female, ovaries, they're sex organs. So they're cells and stuff. So are they X or are they Y?
Oh, they're X and X. So
Just X. But
it's
if
the male.
we're
The
doing,
male
remember,
determines the
XX,
sex.
sorry, XX means you get it from your mother and your father, but we're talking about specifically haploid cells. That's one. So haploid cells are 23 chromosomes. It's either X or Y. Female has what?
X.
Yep.
Female
What about
X.
male?
Yep.
So
male
sperm.
could be X, could be Y.
Yep, perfect. So that means technically the sperm is going to determine the sex. Because if the sperm is X, it's going to be a female because it's going to go XX when they cross. If the sperm is a Y, it's going to be male when they cross with the egg. So this is just something I like to throw out there because a lot of students neglect this information and it makes it hard to understand genetics. I mean, you can go into more detail about meiosis, mitosis. I think you probably should for those obviously watching at home. But it's important we know all of these definitions before we dive into genetics because a lot of words are thrown around that confuses people. And they think that when we say genes and alleles, an allele is a variation of a gene. But when we're saying there's 2 different genes, there's 2 genes, but an allele is a variation of 1 gene. So there's different nuances to understanding what is being told. So we dive to question 1. Would anybody like to tell me, so Monica or Jeremy, sorry, took a bit of a sidetrack there just to make sure that everybody's on board.
One interesting notion is today people who study genetics, they think that eventually, many years, there's a hypothesis that the Y chromosomes are going to actually become extinct. Like it's going to vanish when because the Y chromosome is becoming smaller and smaller. Because only one it's like 1 gene on there that's the only utility of it. So it's got rid of all the other. There's no other material on there that's needed. So eventually, that 1 gene might just hop on to the X chromosome, and so
we're all going to become female. What an amazing world that would be, actually, far out. Because men have definitely destroyed the world today. So probably better.
Whether that's expressed or not.
Yeah.
All right, but for the question, I think so. For the South, I thought the South was a bit more typical of Mendelian ratios. So, like, dealing with quarters and like 3/4 being dominant and then 1/4 being the recessive. So that's where I thought, okay, brown looked like dominant and the white seemed like it was the recessive. So that was theory 1, I believe.
So you've got theory 1. How about you, Monica? I think you got theory 1 as well. Would you like to give your thoughts about this question? You don't have to if you don't want to, if you'd like to.
I'll be honest, I pretty much, just did a process of elimination. and a lot of it was guesswork. So yeah, I would really like to hear what maybe Jeremy or what you've got to say about your
Well,
thought
look,
process.
you got the right answer. So your process of elimination worked, and that's all that matters in the GAMSAT. It's multiple choice. You got the right answer. So what Jeremy had was on the right track. It was correct. And there's actually a very, very easier way. Jeremy touched on it. There actually is a very easy way. So if you think about it, it says in the south of the country. We know that in the south, 3/4 of the rabbits are brown. So I'm just going to throw it out of there. If you've got a population where a majority is going to be a specific phenotype, is that going to be a dominant or a recessive? Most likely speaking, is that a dominant or a recessive, let's say, allele or gene? What would you say? It's dominant or recessive?
Dominant.
So would brown be dominant or recessive if it's 3/4 in the population? Dominant?
Dominant.
Yep.
Yeah.
So you just want to look at the three theories. Which of the three theories has brown coat as dominant? Well, one does. So the wild coat, brown coat is the wild type, so A. Then you've got the recessive is white coat, little a. You've got here theory 2 is white coat is the wild type, which is dominant. And theory 3 is that the white coat, again, is the dominant. So that's the wild type. And there's little other things here. But you can see clearly just by process of elimination, you don't have to look at the other theories and draw all the Punnett squares. You know straight away just by looking at it, because in GAMSAT, ACER likes to create questions like this where if you're really, really switched on in the exam, which is hard because sometimes you're just like, what's going on right now? If you're really switched on, you'll note that straight away it's dominant, which means you're most likely going to be theory 1. And if we do the actual, I've written it down here just before, if we do the cross, so let's do the cross. So you've got a with a little a, and then we've got over here big A again with a little a. So we've got AA, and then 2 little a's. But just notice it says as well, a neutral allele controls for secondary color in the coat, which is activated only in white rabbits. And white rabbits are only here, but it's not essential. So over here, you're going to also have an additional BB. That's going to determine the coat. But it doesn't matter because it says here there's an even mixture of chinchilla, Himalayan, albino. So this doesn't matter. But you can see here you've got, so it tells you brown coat is A dominant. So you've got 1, 2, 3. So you've got, I think I've drawn it here. No. So you've got now 75% brown, and you've got 25%, this one, white. And it says south of the country, it's 3/4 are brown and the rest is white. And because it's got this random BB neutral allele, because it's neutral, you're going to have an equal amount of the rest. So straight away, you know it's theory 1, so you don't have to worry about the other theories. The other theories gets a bit tricky, which we'll go through in the next slide. So just note that theory 2 and theory 3 have no way of accounting because mainly white is the dominant, and you can't have a majority brown if white is a dominant. It's not possible. So that's, I guess, the quick way of looking at it. So number 2. Now, this is a tricky one. Do you have any thoughts on number 2? What are your thoughts here for the north of the country?
Are you asking?
Anybody who'd like to jump in? You'd like to jump in, Jeremy or Monica? You can go. Let me know what your thoughts are.
Well, I just wanted to point out like the 33% for me is what made it really challenging because I just went off like all I could think of was, okay, it's like usually you deal with one allele, you've got four possible outcomes, and then like, you know, it's like 1 out of 4, 2 out of 4, 3 out of 4, and then you can times that, multiply that by another if you have a second allele. So if you're dealing with 2 alleles, then there's like anywhere from 1 to 16, or 15 out of 16% chance of getting, there's like, it's out of 16, and so out of those percentages, I was like, oh, I can't remember when you would get 33%. So that was, or like 2/3.
Yeah, I think I was looking at it again, it's probably just like looking at the options, right? And then read theory 2 and theory 3 and just sort of thought like, well, it's saying that white is the recessive allele, which results in brown rabbits. And then when the second gene is activated in white rabbits only, the dominant allele is albino rabbits, right? So like that's basically saying that it's kind of even, I guess. And then in theory 3, what they're saying is that it's essentially like, I don't know, I feel like it was essentially saying the same thing. And that's why I picked theory 2 and 3 because they were the options. Do you know what I mean?
That's all right. That's
like,
fine.
I don't know if that makes sense, but that's what
No,
went through
look,
my head.
That's your thought process is important here. We need to know what your thought process is, and then we can walk through how we can mold that thought process. I think the trick here is, this is why at the beginning, but thanks for the contribution, Monica. You were almost there. You too, Jeremy. This is why at the beginning I asked if what is the difference between these different terms. So theory 2 is giving you 2 genes. It's saying there's 2 genes involved. So when you do your Punnett squares, you have to take into account 2 genes. We're not just talking about 1 gene with a dominant recessive allele. We're talking about 2 genes here. One has dominant recessive, the other gene has dominant recessive. But it's telling you for theory 2. So the first gene, so you're going to have obviously your A, and your B. So there's different variations of A, and I'll show that in a second with the Punnett square. But just note that it's saying wild type. So A is going to be the dominant, white, recessive is brown. However, if you do have a white rabbit, you also, the variation in whether it's going to be chinchilla, Himalayan, or albino is determined by the second gene. So the way we draw it then is like this. If I just grab my eraser. So now you're probably going to be like, makes sense. And the trick here is we've got 2 genes involved. It's not just your standard. Theory 3 is practically 1 gene with multiple alleles. So you're going to say something, Monica?
Oh, no, sorry. That was an accident. Yeah.
No, I just, because it just popped up. Oh, your microphone came on.
I'm unmuted. No, sorry. That was an accident.
No, you're fine. So you'll see for theory 2, we've got 2 genes. It's not just your, well, I can show you theory 3 as well. You can see that theory 3 is just pretty much 1 gene with multiple alleles. So you've got your dominant, recessive. Whereas for theory 2, you've got 2 genes. You've got a dominant and recessive for A, but then if you do, so A is white. If you get a white, then your B is activated, your B gene is activated to give you whether it's albino, albino, chinchilla, or Himalayan. So let's walk through it carefully here. Theory 2 is saying, remember, it says wild type is dominant A, recessive is little a for brown rabbits. So first of all, let's just look first if it's going to be a white rabbit. So we know straight away if we draw it. So we've got all the variations: big A, little a, big A, little a, and then you've got your big B, little b, big B, little b. So you've got the variations. And remember, this is how easiest way to remember it. So remember what we said: sex cells are haploid, which means you're crossing one parent with another parent. So you've got the genes from one parent and we're crossing it with the genes from another parent, and we're going to end up with, say, a he, B, B, then you're going to end up with A, big B, little b, so on, so on. So that's how it looks, but it gets confusing when you draw it like that. But just keep in mind that when you're doing Punnett squares, you've got it's like you're mixing two haploids together to make a diploid. So these are the genes from one side, these are the genes from the other parent. So A, we know it's going to be albino, so it's going to be white. Let's just say white. We know it's going to be white, big A, white, big A, white. You're crossing here again, it's going to be white. Now, you've got, so because it's all white, remember what it said. If it's white, B, so if it's a big B, so that's the dominant, it's going to be albino. Whereas if it's a little b recessive, it doesn't say explicitly, but if it's little b recessive, it's going to be the variant. So the variant is going to be, it's either going to be, or well, let's just call it the other white. So we don't know what the variant is, but it's the other white, so which is WO. So if we take a look, it's big B, big B, big B, big B. So it's all going to be albino. Now, this is where it gets interesting. So we've got AA, so it means it's going to be white. AA means it's going to be white, but you can see that here is a big B, little b, so this has to be albino, but you've got two recessive little b's. So we know it's going to be white, but at the same time, because it's recessive b's, the second gene, it's going to be a other white. Same thing here. Big A means it's going to be white. Little b, big B means it's going to be albino. You keep going down, keep doing for the rest, and then you're going to find out here, little a, because we know that the first gene tells us if it's recessive, it's going to be brown. So we know straight away it's going to be brown. Over here, little a has to be brown. Same principle here. So you can see when you do the whole table, and this is where the trick is, you just have to draw the Punnett square. You can see that there's how many albinos we have? 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 out of 16 albinos. You've got 1, 2, 3 others. So albino, 3 others, and then we've got 4 brown. So if we do the math, there's about, so out of 16, so 25% brown. So the others is about 19%, and the albino is about 56%. So does that theory account for, so it says here the north says there's 33% each. So clearly, theory 2 is not correct. Theory 2 is not correct. So let's see theory 3. So before I dive into theory 3, does that make sense, guys and girls? Is that I know it's hard to follow straight away, but do you understand because we're using 2 genes?
Yeah, that makes sense.
How about you, Monica? Is it okay, or?
Yeah, no, that makes sense now that you've explained it.
Yeah, I know it's hard with genetics. It's one of those, this is where the trick comes in, because I've seen GAMSAT questions in the past where they trick students by including 2 genes. This is where they trick students, because yes, you've got a gene for a specific color that might have a dominant and recessive allele, which is a variation of a gene, but they might incorporate multiple genes, which tricks people, because you can have one gene with dominant recessive and you can have another gene dominant recessive. And then when you cross, the key here is when you're crossing, remember you're crossing haploid sex cells, right? So you're crossing one partner. So let's say this is male and this is your female. So remember sex cells, remember we said 23 chromosomes. And in this instance, we're using 2 of the genes from the 23 chromosomes. Whereas over here, this is our male and female. Now for theory 3, we've got just one gene with multiple alleles of the same gene. That's important to know. It's not like we've got 2 genes, 2 different genes. It's one gene with multiple variations of that gene. That's the idea. And it's telling us here, for the wild type, it's white, and there's a recessive for brown, and also another recessive for other alleles. Now, we don't know how white replicates develop further variation, so we just know they're white. So if it's if it's anything that's a recessive, it's going to be brown. That's the idea. So you know straight away if you do A, so we've got a gene with 3 variations. So we've got the dominant and 2 recessives. I know that for the rabbits, I think it goes up to, I think, 4 or 5 different alleles. So you never know, you could get something similar in the GAMSAT. But when you draw the Punnett square, you'll see that it has white, dominant, 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 white, 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 dominant, white. So everything recessive is going to be brown. So you see, therefore, theory 3 is going to be 1, 2, 3, 4, 5 whites to 4 brown. So, and that comes down to which one? So which one, therefore, do you think theory 3 matches? So the answer for 2, so you can see clearly that the answer for 2 is none of the theories, because for the north of the country, you have to have 33, 33, 33. Over here, we just know it's white. It's white 5, brown 4. So it's just slightly more. So 2 is none of the theories, but 3, sorry, theory, question 3, what do you reckon the answer is then? Remember, so it's 5 white.
So for the last one, for the last question, what's interesting is like, if you just ran it up, like you could go, I mean, like you could go, okay, 5 out of 9 is close to 6 out of 9, and that's 66%, and then which can include all the other white rabbits. So you can kind of make it work if you start changing the numbers, but then if you keep it like the exact numbers, there's no real, it doesn't really, yeah, it's not the classic Mendelian ratio, like the Mendelian, like the percentages that are, you know, I haven't come across this before where there's 3 columns. So
yeah, because there's different variations, because you're used to, so this is what we mean at the beginning where we said it's a very gross oversimplification to think that we only have 2 alleles. In humans, generally we do, and obviously in trisomy, you've got 3 different variations of a chromosome. But in rabbits, I think it's gene C, it could have up to 5 variations of a gene, so 5 alleles. So it's amazing, isn't it? So, and this is the sort of thing that I love asking you in the GAMSAT, stuff that's going to throw you off and be like, I've never seen this before. Hopefully now you've seen it and you won't throw you off.
With the third question, I just felt like there was not enough information in somewhere. I was like, okay, how much more is slightly more? You know, like, is it, if you're going, what did it say? In the center of the country, there are slightly more white rabbits compared to brown.
Does this look like slightly more? In
well,
2 variants.
Yeah, I mean,
so the middle of the country.
I mean, theory 2 and 3, there's more white than brown, but yeah, again, I was like, oh, what do I do? If it's a normal, first I started with because theory 1 suited the south, and then I thought, oh, in the center there's slightly more. Okay, well, instead of you know, 3 out of 4 brown, 1 out of 4 white, then maybe there's 2 out of 4 white, 2 out of 4 brown. But no, that's not slightly more. It would have to be the reverse: 3 out of 4, 1 out of 4 brown. But that wasn't really, I guess, yeah, looking at it now, that wasn't an option. So yeah, that's why I said none of the theories. So I think it's because I didn't do this, I didn't draw this out. That's why I thought none of the theories, because I didn't realize I could draw the 3 columns in the moment.
Do you have any comments, Monica? I think you're still on. Yep.
Sorry. Yeah, like I said, yeah, I don't know. I really just guessed this one. yeah, I'm not really sure.
No,
Sorry.
that's all right. Do you feel as though you're better off now if you get a very, because you know how A series, you've sat the GAMSAT before, they love to chuck questions under time pressure that just throws you right off.
Yeah,
Yeah.
so that's why I sort of tried to do like a semi-educated like guess based on the options that they give you. And yeah, like, I mean, it's been a long time since I sat it, but I just found that like that was the best way to get through it, kind of thing. So you do have to know a little bit, but then eventually it is just like trying to find out the information from what they've given you.
The good thing is in the GAMSAT, the good thing is a lot of you with your educated guessers got correct answers. I think the first couple, which is fantastic because that's all you're asking in the GAMSAT. We just have to, because you're not going to know everything. There's questions designed to throw you off. The key here is, which you guys have done correctly, guys and girls, correctly, is that if you feel as though it's difficult, all you got to do is just take a calculated guess and move on, which you've done, and you've got points out of it. That's the best part. When the thing is with GAMSAT is, if you don't know the answer, the next best thing is to take a calculated guess and get as much points as you can and just run, which you've done, which is fantastic. This question, yes, the answer is theory 3, because it's saying, I think the important thing here is it's saying best explains the population. So out of the 3 theories, definitely theory 3 best explains the center of the country, because there is slightly more white than brown, and we don't know if it's chinchilla, Himalayan, albino, or whatever, but we just know that there is slightly more. So technically, it does fit the bill. Sorry. So that would be correct. Yeah.
I've made a discovery for myself. Yeah, I didn't read it properly. So
no, you're fine. It's normal in the GAMSAT. It's going to happen.
I was trying to think of again what theory would, like, what Punnett square would it look like? But if I just focus on the question, going, oh look, if it needed more information, there might likely be a multiple choice answer saying unable, like needs more information.
Yeah.
But because it's none of the theories, it's like you're saying, you're just focusing on the theories here, and then I would go back to the question and go, oh, it's actually saying the best fit, not what it, yeah. So just like, what's the best sort of theory, the most, the most correct.
Yep.
So
that's good. Yeah. That's good. This is very good, because we've got 9 days until the GAMSAT, so there's still plenty of time to make sure that we rub these out of your system, so that now that you do get in the GAMSAT, I can guarantee you're going to make sure you read it.
If I get a question and I spot those key words like the best explains, best fit, I'll be like, all right, just it's could be one of the answers here, right? And again, look for the wording. If it says not enough info in the answer, then maybe there isn't enough info. But if there's no answer that says needs more information, then yeah.
So just to summarize this for those watching here and at home, just remember, if humans are diploid, but our sex cells, our gametes are haploid, which means when we're crossing for our Punnett squares, you got to make sure you're crossing. If it's specifically saying, like, for example, theory 3, it's for one gene, right? It's telling you we've got a gene. This is one gene. One gene has a dominant, and it also has recessive types of that specific gene. So you've probably inherited, so you've got a dominant here, you've got a recessive, and you've got another recessive. It isn't just two, obviously you're going to have big A, little a. We've shown you in rabbits and other mammals, there could be multiple alleles of one gene. However, the question is only talking about one gene here. Look at theory 2. Theory 2 is saying you've got one gene, but then another gene is activated if something else happens. So you have to remember you're going to be crossing, let's say, another gene. It could be big B, little b, or whatever. So this is one gene. Now we've got introduced another gene. That's for one one person or male, female, and then you're crossing it with another male and female. Once you can wrap your head around this, I swear, any genetics question you get in the GAMSAT is going to be like that to you, because once you can wrap your head around what an allele is, what a gene is, what the difference is between the two when you're doing Punnett squares, you'll become a pro. Now, I guess that is the end of today's webinar, but I think there's a few, the GAMSAT is in 9 days, and I think the best advice I can give you before you sit the exam in 9 days is to relax. You do have, now's the time to start focusing on your mental health, because if you stress yourself out from now until whenever your exam is in the next 9 days, 10 days, you're just going to do yourself a disservice. I always say now's the time to keep writing, typing, stimulate your brain. Now's not the time to cram stuff that that you haven't studied before. You have to consolidate your own study that you've done in the past. Learning new things is not going to be productive, because I mean, if you've had 6 months to prepare for this GAMSAT from, say, September. If you're cramming now, it means you haven't prepared in the last 6 months. So try to keep your brain fresh so that when you sit the GAMSAT, you're not getting the questions you should get right, you're not getting them wrong. So let's consolidate that. Keep writing every day, write about anything. Keep typing, well, if you're going to be doing your section 2, and just actively read things. And if you can do that, de-stress, go for a run, go to the gym, get your mind off study, because the more you relax yourself, and just remember, you can do the exam as many times as you want. You don't, and in Australia specifically, you can use your GAMSAT score for 4 years now. So unfortunately, UK and Ireland, it's still 2 years, but in Australia, you can use your exam score for 4 years. So you've got plenty of time. Don't stress too much. Is there anything else, any other tips you'd like to pass on, Jeremy and Monica? I guess in your GAMSAT journey, how you'd prepare, or how you'd prepare yourself for the exam?
I guess you prepare. I mean, last time I did it, it was online. So there's a bit of a novelty this round again for me. Second time will be the first time in person. So I'll probably learn things this time round, but what I learned last time was, just just making sure you're prepared well early, because in my experience with normal exams, like I've in the past I did a maths unit and I didn't have a calculator, and it was in summer, and I ran from one end of campus to the other because I had to get this calculator, and then I ran all the way back to the exam hall, and it was so embarrassing. I got there, I was sweaty, and I learned my lesson. Just what you want to do is just get there early, and just kind of take that time, that half an hour beforehand, to just like meditate, relax, and so preparing. But this, the recent the webinars, that work. It's been nice because I've got this, I've done the prior learning of the theories and stuff, but doing these practice questions, it's taught me to take another look at my the exams, the exam skills, rather than just the science skills. Like, don't forget to eliminate the least likely options, and rather than don't forget that sort of stuff. If it's not coming to you straight away, those little things count. I think because I thought I was pretty competent with the genetics. I then didn't use those exam skills, like those strategies where you're doing elimination strategies, or you're doing like in the best fit. So kind of just quickly going, before I go to the next one, can I just eliminate the other two and
Mm.
yeah.
No, it's good to hear. How about yourself, Monica? Do you have any comments to add of how you're going to prepare, or are you just letting the stress ride?
Yeah, no, I think basically what you said, just to not stress too much, but I think at this stage it's more just about practicing, like sitting there for long periods of time, concentrating, not stressing too much, or else then just gonna like psych myself out for the actual day. But yeah, it's pretty much just doing some practice questions, getting my technique down pat as much as possible, and maybe just a few essays just to like practice writing them. So yeah, it's going to be a long day, but I think
I definitely will be. You'll be sitting there for a while. My goodness, it's
yeah.
very long day. I think that's the big one. Mentally preparing yourself to be sitting in a room all day stressed. It's exhausting. The mental fatigue is real. For those who've sat the exam before will know mental fatigue hits you, because when you get up to, like, say, section 3 towards the end, you're just like, you have a mental block. So you got to time the time you drink your water, your meals, when you eat your food, what foods do you eat, nutrition. You just got to make sure you don't psych yourself out, as you said, Monica. And for those who do have clinical anxiety or depression and stuff like that, obviously we're not trying to simplify the illness. If you do have a problem, please do seek help, because there is help out there, and you can help yourself. You can get help, because if you find out that, through GAMSAT, you do have some form of clinical anxiety, please help yourself as soon as you can, because you don't want to get to the point where it ruins your life. And there is help out there. For those who do have stress induced from the GAMSAT, which is synonymous with anxiety, but let's just say stress, definitely, as you said, Monica, practice exam pressure, because it does hit you hard like a train or a bus. So look, you do have 9 days. It's great to have you guys today, guys and girls, in today's class. I think it was definitely beneficial. If you do want to watch the recording, if you're on the mailing list, I'll send it to you next couple of days, and it will be posted on YouTube soon. You can contact us via email or visit our website, follow us on social media. And we'd love to help you. Is there any last questions before we sign off?
No, I think that was really good. Thank you so much.
No worries. Thanks, Monica. How about you, Jeremy?
I enjoyed it as always. Thanks, Hassan.
No worries. Thanks, guys. Have a fantastic weekend and good luck in your exams. And if you want to have a debrief or anything, call us for a free consult. We can discuss. Obviously, GAMSAT applications are coming out soon, and university applications are coming out. We can definitely help you, and we'd love to help you. Take care. See ya.